So in the last class we were discussing about sex determination. In sex determination we concentrated what is sex determination. In that we studied two technical words. One is called heteromorphic. Heteromorphic means the gametes or the organism produces or the sex chromosome produces two different or two dissimilar gametes. So they are called heteromorphic or heterogametic. And second one is homomorphic. In the sense, sex chromosome produces or sex chromosome forms one similar gametes. So that is called homomorphic. And these gametes are called homogametes. And the second part that we have discussed is how the sex is determined in human beings. So there are two set of chromosomes we have discussed. One is the autosome and another one is the allosomes. Autosomes are body chromosomes and the allosomes are sex chromosomes. So how this is involving in determining the sex that we studied. Before that we discussed what is heterogametic male and heterogametic female. Then we also concentrated about the importance and the structure and functions of the Y chromosomes. So these are all very important in the last part that we have discussed. And today we will be concentrating on another topic in this called dosage compensation. Compensation, you know, something that is reduced. So you are compensating that reduction part of it. Okay. So since we are concentrating on sex determination, this compensation we are going to study based on the chromosomes. So we are going to compensate the chromosomes. Now, when you talk about the male as well as the female, in both, the chromosomes must be equal. The number of the chromosomes must be equal, or the gene which is presented inside the each chromosomes must be equal to for both male and for female. For male and female, the chromosomes must be equal. Clear? Right. So now, when we talked about two different chromosomes, that is the autosome as well as the allosomes, among these two chromosomes, male and female will have equal number of autosomes, that is 44 chromosomes. Both male as well as female, autosome will be always equal. When you talk about the allosome, we discussed two things, that is the X chromosome and Y chromosomes. Now, we also studied the structure and the gene which is presented between these two chromosomes, that is the X chromosome and Y chromosome. X chromosome, the size is 165 MB, you remember? And Y chromosome size is? Very good, 60 MB in size. And how many functional genes are there in the X chromosome? 1000 functional genes are there in one X chromosome. And how many functional genes are there in the Y chromosome? 60 functional genes are located. So now, when male is having one X and one Y, female is having X and X. In these two chromosomes, in this male as well as female, the autosome only is equal, but see the allosomes, male will have only one X chromosome, only 1000 functional genes are there. But female, two X chromosomes are there. That means one X chromosome will have 1000 genes and another X will have another 1000. So 2000 functional genes are there. So that means the allosomes are not equal. Only the autosomes are equal. But what we are going to do is, if this condition is there, you cannot have an equal number of chromosomes. So what we are going to do is, we are going to compensate this dosages in this number of the genes. Okay, so every chromosome in male and female must be equal, including the shape and size, including the number of the genes which is located inside the chromosome. But what happens in the sex chromosome? The number of genes varies. than the male gametes. So what we are going to do is we are going to compensate this. How we are going to compensate this? Now female has 2 X chromosome. This is for the female. She has 2 X chromosome. What we are doing is we are going to make 1 X chromosome inactive. Okay? So this is physically made. Now among these 2 X chromosome, 1 chromosome becomes inactive. That means not functional. So every female will have only 1 functional gene, functional chromosomes. Only 1 functional. So out of these 2, 1 becomes inactive and another becomes active. So here the dosage is getting compensated. Why? Because male is also having 1 active chromosome, female is also having 1 active chromosome. And this inactive chromosome, I told you, it is not functional. It is not going to do any function at all. So we call this as heterochromatin. Heterochromatin means there are 2 set of chromatins that we studied when we talked about Y chromosomes. So that is euchromatin and heterochromatin, non-combining region of the Y chromosome, NRY region. You remember? Heterochromatin, non-functional. Euchromatin as a functional gene called SRY gene, which determines the testicular development factors. Same here, this inactive chromosomes are called heterochromatids. Now how this heterochromatin looks like, it is a tightly coiled, looking like a tightly coiled structure which is present inside each cell. Okay, so this one is a loosely coiled. Why? Because it is functional. Okay, all this functional, you know how it is going to make another progeny, everything that we will concentrate when we study about genetics, 5th chapter. But how it looks, inactive chromosomes are tightly coiled. So it becomes heterochromatic. And euchromatins are always loosely coiled inside. Is that clear? This much? Right. So this is how we are compensating the dosages in the chromosomes. So now I just come back. The chromosomes must be equal in male as well as for the female. But when you talk about the autosomes alone is equal, not the allosomes. Why? Because male has 1 X chromosome whereas female has 2 X chromosomes. In the sense, female will have more number of functional genes compared to the male. So you have to compensate that dosages. So how it is getting compensated? One of these X chromosome is become inactive to form heterochromatids. And this was developed by Mary Lyons. So this is called developed by Mary Lyons. So this theory or hypothesis is called Lyons hypothesis. Okay. Now we called this 1 chromosome which is inactive is called heterochromatic, which is tightly coiled, and we also call this as bare bodies. So very important 2-mark question, bare bodies. What is bare body? Inactivation of 1 X chromosome. This much you are clear? Right. Now, this bare body was found in the nerve cell of cat. Nerve cells of a female cat. Not in the male cat. Why? Because bare bodies are present only in the X chromosome, not in the Y chromosome. In Y chromosome, we have only 1 function gene called SRY gene. That's it. But whereas the bare bodies are present only in the X chromosome, so it is found only in the female. So this was identified in the year 1949. Which one? Bare bodies. From where they have identified? In the nerve cell of the female cat. So it looks like a tightly coiled structure which is presented inside the female cat. Is that much clear? Right. Now, you know what is bare body? What is bare body? Inactivity or inactivation of 1 X chromosome. So, if this bare body is given, an application oriented 1 marks will be asked for you regarding this bare bodies. There is a rule for bare body. that is n minus 1. This is a formula for calculating the bare bodies. N minus 1. Here n talks about the number of X chromosomes which is presented in each cells. Okay? N is for the number of X chromosomes and 1 is for the bare bodies. So how you will calculate the number of bare bodies? That is n minus 1. Total number of chromosome, X chromosome minus 1. So suppose if female is having 2 X chromosome, what is the number of the bare body? How? 2x minus 1. So bare body is 1. If the female is having, XO type, how many bare bodies are there? How many bare bodies are there? XO type. Well, very good. There is no bare body at all. No bare body at all. Suppose a male who is having X and Y. How many bare bodies? Huh? No. 1. I told you that there is a rule for it. There is a formula. N minus 1. Total number of the X chromosome minus 1. Here the total number of X chromosome is 2. So 2 minus 1. So the total number of bare body is 1. Is that clear? So this is how you calculate the total number of the bare bodies. So that means how you will calculate, because the number of the bare body is 1 less than the number of the chromosomes. The number of bare body is 1 less than the number of the X chromosomes. This much clear? Right. So that is how we talked about the dosage compensation. In dosage compensation, we are going to concentrate on the allosomes, where there is not equal number of genes presented for male and female. Why? Because female is having 2 X chromosomes. One X chromosome will have 1000 functional genes and another will have only 60. So that the sense male will have only 1 X chromosome. Here it is 2 X chromosomes, so together it will have 2000 and they will have only 1000. So naturally 1 X chromosome is become inactivated, so that it becomes heterochromatic. Always heterochromatics are non-functional. So if it is non-functional, it should be tightly coiled, tightly packed inside the cells, inside the nucleus. Where this chromosomes are, it is there inside. These genes are presented inside the sex chromosome, which is there inside the nucleus. This is a cell. This is a cell. Inside this nucleus only, these chromosomes will be located. So inside this, it is tightly packed and kept inside. And whereas this is loosely packed, that's all. It is kept inside the nucleus. One part is tightly kept, one part is loosely kept. So that loose part is called euchromatin. Euchromatin is going to do function. That function at all we will study detail when we study about the fifth chapter. Is that clear? Right. So now what is the other name for this inactive chromosome? It is called barren. So Mary Lyon only suggests this one. So this hypothesis is also called as Lyon's hypothesis. But who has discovered this first nerve cell in the bare bodies in the nerve cell? That is in the 1949. First time is given mark, 1 mark question. Barr and Batten is an important 1 mark question. In 1949, Barr and Batten have discovered this bare bodies in the nerve cell of female cat. It is not presented in the male cat. Why? The reason is bare bodies are only presented in the X chromosome. So it is belong only to the female. So it is absent in the male. This much clear? Right. Just go through for 2 minutes. So the next topic that we will discuss is last part of this topic, sex determination. That is haplodiploidy. And another important 3 marks, sometimes it is asked 5 mark question, book back question too. Right. So now, when we talk about haplodiploidy, 2 things that is being focused. One is called the haploid, another one is called the diploid. You know what is haploid? Haploid means, haploids are half a number of chromosomes. So n number of chromosomes are called haploids, half set of chromosomes. And what is diploid? That is 2n number of chromosomes, full set of chromosomes. Now, we are talking this haplodiploidy under a topic called sex determination. Okay. So, in some organism, half a set of chromosome decides one kind of sex, male or female, and full set of chromosome decide another sex, male or female. So that is the meaning of studying haplodiploidy in sex determination. So now we will come back to this in a family or an order called Hymenopteron order. This is an order where some insects comes under this order. We studied animal kingdom, starts from phylum and ends up to the order. So certain insects comes under an order called Hymenopteron. Okay. So what are all these insects comes under these Hymenopterons are ants. Second one is the wasp. And the third one is the honeybees. So now these three things are insects comes under an order called Hymenopteron. Now in this insects family or Hymenopteron order, the sex has been decided, that is male or female is decided by the number of chromosome it receives. In Hymenopteron order, male or female sex is decided by the number of chromosome it receives. For example, in this family, if it receives single number of chromosome, that is half a number of chromosome, haploid number of chromosome, it forms maleness. Suppose if it is receiving 2n number of chromosome, full set of chromosome, it becomes female. So, number of chromosome decide the sex in this Hymenopteron family. If it is diploid, it gives you female. If it is haploid, it gives you maleness. That is what haplodiploids are. This much you are clear? Haploid means n number of chromosomes, half a number of chromosomes. Diploid means full set of 2n number of chromosomes. So in Hymenopteron family like ants, wasps, honeybees, if the female receives haploid, that means half a set of the chromosome, it forms male. If the female receives full set of 2n number of chromosome, it becomes female. That much you are clear? Right. Now we will talk about, in your syllabus, other two things are not given, but honeybee is given. In honeybee's family, the sex is decided or determined by haplodiploid. That means the number of chromosome it receives. Okay. Before we could go for this content, we will talk something regarding the honeybees. Now when you talk about the beehives, what is beehive? You know, beehives. In that beehive, you will have three varieties of honeybees. Number one is, you will have the queen bees. You will have the queen bee. And the number of the queen bee is only one. In one beehive, you will have only one queen bee. Understand? And this queen, naturally it is female and it always possesses 2n number of chromosomes. Why? If it receives 2n number of chromosomes in Hymenopteron family, in order it becomes female. Okay. And the function of this queen bee is only to lay eggs. That's it. Only to lay eggs. This much clear? Right. Now the second one is the worker bees. The worker bee is many in numbers, many. This is also female. It also receives complete set of chromosomes because in Hymenopteron order 2n decides female. This much clear? Right. So that means workers are many, female 2n number of chromosomes. The role of this worker is go out, collect nectars and come back. And another important is it will raise the or nourish the eggs that is laid by the queen bee. It will look after it. So it is called kin selection. Kins. You know what is kith and kin, no? Kith and kin, your family relatives. So that means here it is going to do the kin selection in the sense it is going to nourish that egg laid by the queen bees. This much clear? Right. And the third one is the drones. Drones are called male. Male are called drones. They are many in numbers and this is only half a set of number of chromosomes, not fully. So in honeybee's family, half set means definitely it will be male. If it is full complete set of chromosomes, it will become female. Understand? Right. So now here, the main function of the drones or the male is only to fertilize the queen. That's it. Okay? Only to fertilize the queen. So this is the actual bees which is located inside the beehive. There are three types of bees which is located in the beehive. One is the queen bee, worker bee, and the third one is the drone. Clear? Right. So now this queen bee will start to lay two types of egg. One is called fertilized egg and another one is called unfertilized egg. There are two types of egg laid by this queen bee. One is called fertilized and another one is called unfertilized. Now see, this queen bee after mating with the male partner, the sperms are received by the queen bees. The male donates the sperm and the queen receives the sperm and keeps inside the structure called spermatheca. Spermatheca is a structure where sperm cells are stored temporarily in the queen bee or the female. Okay? As soon as the sperm comes out of the male, the sperms are stored inside the spermatheca structure where the sperm cells are stored temporarily inside the female reproductive part. Now, this queen bee, it will send an egg and allow the egg to fuse with the sperm. If this egg fusing with the sperm, it becomes fertilized egg. Okay? And the queen bee will not allow some eggs to fuse with the sperm and just lay the egg. Understand, before laying the egg, the queen bee makes some eggs to fuse with the sperm and get fertilized and lays fertilized egg. At the same time, it will not allow certain egg to fuse with the sperm and just releases unfertilized eggs. Understand? So always the fertilized egg becomes female. Always the unfertilized egg becomes male. This much you are clear? Clear? So every time when the queen is laying the egg, there should be two types of eggs. One is called fertilized egg and the one is called unfertilized. So now who is deciding the sex of the honeybee's family? Queen, as well as the number of the chromosomes it receives. Now you understood how queen bee is deciding the sex of the individual. How? If it is making the egg cell to fuse with the sperm cell, it will get fertilized. If it is not allowing the sperm cells, egg cells to fuse with the sperm cell, it just lays the unfertilized egg. So that's how the queen bee is deciding the sex of the individual. Okay. Now what we are going to study is how the number of chromosomes is going to decide the sex of the individual. Is that clear? This much you are clear? Right. Shall we go for the other part of it? Now, here, when we talk about the number of chromosomes it receives, I'll just come back here. Okay. So now, when you talk about the number of chromosomes, first we are talking about the queen bees. Queen bee, the number of chromosomes is diploid. The number of chromosomes is diploid. What is the role of this queen bee? it starts laying eggs. Okay, egg is laid. Now listen very carefully. The queen is 22 n means it is having 36 number of chromosomes. Okay, honeybee will have 36 number of chromosomes as we have 46 number of chromosomes. Now, egg is a gamete. Egg is a gamete in the sense when this queen is going to make egg, this 36 chromosome must be undergo reduction division to form 16. Haploid 16. So this division is called meiotic division. That is what we studied in the spermatogenesis and oogenesis. You clear? Right. So in spermatogenesis and oogenesis, what happened? Primary spermatocyte, primary oogenesis or oocyte has undergone meiotic division to form haploid because those are diploid. Forms haploid. So the number of the chromosome is getting reduced. So meiosis are always called as reduction division. So what is happening? When the queen bee is going to lay eggs, this number of the 36 chromosome is getting reduced into number of 16 chromosomes. Okay. Now, when you talk about the male partner, the male total number of chromosome is 16. Why? Because it receives only half the number of the chromosome. It is only 16. So now when the male is going to form gametes, what is the gamete formed by the male sperm? When this is formed, if it is undergoing reduction, it becomes 8. So 16 and 8 will not give you any individual. It should be either half of it or it should be double of it or equal. Okay. Either it should be 16 and 16 or it should be only 16. Either it should be 32 or 16. But what is happening here? If it reduces, it does not become 32. It does not remain 16. Why? Because 8 is here and 16 is there. So naturally what is happening? This 16 will not reduce. It becomes 16 itself by a division called mitotic cell division. We studied what is mitotic. Equal cell division is called mitotic division. Reduction division is called meiotic division. This much you are clear? Now, what is happening during fertilization? I told you this honeybee, what it will do after receiving the sperm cells from the male partner, and it will allow certain cell to fertilize. Now, this egg cell is getting fertilized with the sperm. So when it is getting fertilized, it forms 16 plus 16, 32 number of chromosomes are formed, diploid in nature. Clear? So that means female and male joins together and form the zygote, and in the zygote you have 2n number of chromosomes, that means 32. It becomes male or female. It becomes male or female, 32. It is female. Why? Because complete set of chromosomes has come there. So that means this fertilization, egg becomes fertilized now. The queen lays egg, fertilized egg. This fertilized egg has 32 number of chromosomes. When this fertilized egg hatches, it becomes female. That is, either it becomes queen bee or it becomes worker bees. Why? Both are diploid in nature. This much you are clear? So that means this kind of a reproduction is called sexual reproduction. Why? Because egg cell, sperm cell fuses each other and the egg becomes fertilized, and the queen bee lays the fertilized egg, and the fertilized egg after all hatches and forms the queen or worker. And how this, both are female, but how the difference between queen and worker, how it is formed? It is because of the food that is available in the hive, beehives. Inside the beehive, if it is given a jelly called royal jelly, it is present inside. A royal jelly is present inside. If it eats, if all this female, any one eats the royal jelly, it becomes queen bee. Those who have eaten the bread which is outside, where you will have this honey and the bread will be there, that bread if it is, that is called bee bread. If it is the bee bread, it becomes worker bee. Okay? All female depend upon the food, it becomes queen and the worker. This much you are clear? Right. So that means due to sexual reproduction, queen bee and male together joins give you only female, that is queen bee or the worker. This is clear? Right. So now, this is one type of egg, fertilized egg. I already told you, queen bee lays two type of egg. One is fertilized, another one is unfertilized. So that means, after all this egg formation, production, division, you will have 16 number of chromosomes. Now, this 16 number of chromosome, no fusion of the male. It is not allowing the sperm to fuse with it. So, it becomes unfertilized egg. And this forms male. Why? Because it is having only half, 16 number of chromosomes. Okay. So, now, queen bee produces male by a reproduction, queen bee produces female by the reproduction called sexual reproduction. The egg is fertilized egg. Queen bee produces male by unfertilized egg. That means there is no male partner. So this kind of a reproduction is called, I'll write it here, parthenogenesis. Parthenogenesis means without fertilization, male partner is produced. Same thing you will study in the botany also, parthenogenesis. Okay, without flowering, fruit form. Now here, without male partner, egg itself will be developed into another male called parthenogenesis. This much clear? So that is what haplodiploidy in sex determination. In haplodiploid sex determination, if it receives half a number of the chromosome, it becomes male. If it receives diploid, that means full set of 2n set of chromosome, it becomes female. Understand? So that is what haplodiploids are. Now, two topics we have completed in this chapter. One is called ABO blood grouping, RH factor. Second one is called sex determination. In sex determination, we studied what is sex determination, heterogametic male and heterogametic female. Then also we studied how the sex determination is happening in the human being, and Y chromosomes, then bare bodies, and finally we discussed about the haplodiploidy. Clear? Right. Go to. So now we'll concentrate in another topic, that is sex-linked inheritance. So far we discussed ABO blood grouping, then RH factors, and second we talked about the sex determination, and the third topic in this lesson is sex-linked inheritance. Now before we could study what is the sex-linked inheritance, we have to understand certain technical terms here. Now, see, sex-linked inheritance. You know what is inheritance? Anything that is transmitting from one generation to another generation. Here we are talking about a trait or a character. If you talk about a trait means character. Okay. This trait or character is getting transmitted from one generation to another generation in the same family. Okay, so now this particular character is getting transmitted from one generation to another generation. That means this trait is decided or determined by the genes which is presented in the chromosomes. Okay, so genes are responsible for determining the traits that should get inherited. In the sense, the genes are responsible for transmitting of the traits from one generation to another generation. Okay. Now, that is what inheritance are. So, for this inheritance to take place, you need the genes. Without gene, there is no inheritance. Okay. Now, these genes are located in all the chromosomes. In 46 chromosomes, the genes are located. Now, the genes which is located in the autosomes, in 44 chromosomes, are not responsible for doing this inheritance. So what do you mean by this? The genes which are presented only in the sex chromosomes are responsible for this inheritance. Okay. So these genes, where it is located? Inside the sex chromosome, that is XX or XY. So that is why, since these genes are located in the sex chromosomes, it is called sex-linked inheritance. How this character are getting transmitted? Only by the genes which are located in the sex chromosomes. Without the sex chromosome, without the gene which is located in the sex chromosome, will never get transmit the characters. So the genes are responsible, which is located in the sex chromosomes, are responsible for transmission of the traits or the characters. That is what we are going to study in this chapter, that is sex-linked inheritance. This much clear, right? Now here we got two sets of chromosomes, one is male chromosome and female chromosome. Most of the time, sex-linked inheritance are done by the X chromosomes, not by the Y chromosomes. So most commonly here, the sex-linked inheritance are passed by the X chromosome, and most commonly the male or become the receiving the traits. Male receives the traits, but who's passing this? Female is passing. So female donates or female passes this inheritance through her X chromosome to the male. That is why how male receives? Because male receives one pair of the X chromosome from mother and one pair of X chromosome from father, Y chromosome from father. So here this X chromosome comes from the mother. So mother is responsible for transmitting any traits to the male through her X chromosome. So most of the genetic disease, end of the chapter we will be studying genetic disease. So even the characters or even the genetic disease are transmitted through the X chromosomes, not by the Y chromosome. So all the female are responsible for transmitting the genetic disorders through our X chromosome, and all the male are being affected with the genetic disease. Female carry the disease, female spread the disease to the another generation, and poorly all the male gets affected because of this. Understand? Alright. So now that is what these things we are going to focus on, this content, that is sex-linked inheritance. I hope you understand what is sex-linked inheritance. The gene which is located inside the sex chromosomes, especially if it is located in the Y chromosomes, definitely it is going to transmit that characters. Even some genes located in the Y chromosome will also be getting transmitted. But since this Y chromosome, if it is having any trait or anything that it is having, it will transmit only to the male partner, to the son or whatever. If the father is having a kind of a disease in his Y chromosome, the father will spread to the son, not to the daughter. Why? Daughter will never receive Y chromosome. Poor male always receives Y chromosome. So often female are responsible for transmitting the characteristic or the any genetic disorders that we will study when we come to the genetic disorders. At the timing, you remember, the transmitting of the traits is through X chromosomes. Mostly, one or two, very limited, it is caused by the Y chromosomes. Okay, so X chromosomes are responsible, and more commonly male are being affected by this or getting the characteristics. Right. Now, we'll talk what is sex-linked genes. You know what is sex-linked inheritance. Now, sex-linked gene means you are talking about the gene which is located inside the sex chromosomes. Now, what is X-linked gene? That means in X chromosome, this is X chromosome, will have so many genes inside. All these genes belong only to the X chromosomes. And it is not belong to any other chromosome, only belong to X chromosome. So all these genes which belong to the X chromosomes are called X-linked genes. And these X-linked genes will not have any corresponding towards the Y chromosome, because Y chromosome and X chromosomes are entirely different. That's what we study, except the PAR region. Only the PAR region will get synapsed, will be joining each other during meiosis. And other regions, or all these regions, are called non-homologous region. Homologous means same. That means it is having non-homologous region. So that means these genes will not have any corresponding towards the Y chromosome. Entirely both are different. The genes which is located on X chromosome do not have any pairing with the Y chromosome, because everything is is different there. Same, when you talk about X-linked gene, the genes which is located only in the, Y-linked gene, the gene located only in the Y chromosome, will not have any corresponding region towards the X chromosomes. Is that clear? Right. So now, we call this Y-linked genes or otherwise called as Hollandric genes. Very important, look at 2 mark question, afterwards you study. See, when you talk about Hollandric, hollow means the Greek word, holos means whole, complete. Andros means andric, there we are talking about andros, holos, andros, Greek word. In holos means complete, andros means the gene presented only in the male, andros means male. Androgen, remember, hormone, belong for male. Same, andros for male, hols for whole complete genes. So that means the gene belong only to the male chromosomes or called Y-linked gene or Hollandric genes. This much you are clear? Right. Now, I told you why male becomes more commonly being affected, because male chromosomes are hemicycles. What we mean by hemicycles? Hemi means half. Different. It is having only one X and one Y, hemicycles. That is the reason male become most commonly being affected by this kind of an inherited traits and all. So this is the basic thing that we have to learn in this X-linked inheritance. Now in this X-linked inheritance we are going to talk about how certain traits are passing through X chromosome and how certain traits are passing through Y chromosome. So today we will take one example. We will first complete the X chromosome and then we will go for the Y chromosome, because Y chromosome only one small part we are going to discuss, that is of 3 mark question, and that's all. Okay. So we are completely will focus now on X chromosome. That too, today we will concentrate only one disease that is being transmitted. Since we are talking about inheritance, traits, everything, but here we are mainly focusing on not the characteristic. For example, only I told you traits and all, but what we are focusing is how a particular disease is getting transmitted through X chromosome or through Y chromosome. If female has a disease, female will spread the disease through her chromosome to the male. Okay, so female are always carrier of the disease, or the female are responsible for causing any genetic disease, but male only will get affected. Okay, so rare cases only Y chromosome, that is what we will focus later on. This much you are clear? Right. So now we will concentrate one particular, there are 2 diseases we will be talking about in your syllabus, X-linked disorders. So in that 2 diseases, hemophilia and color blindness. So now in this we will talk about color blindness and we will complete today, and in the next class we will be discussing on the rest of the content. Okay, right. So now we will talk about this X-linked inheritance. In X-linked inheritance we are going to talk about a disease called color blindness. Color blindness. Now what is this color blindness? Actually it is a disease caused by X-linked disease, X-linked disorder. Why? Because X-linked is female is responsible for carrying this disease. Female is going to carry this disease. Okay, right. Now, to whom she will spread this disease? She will spread the disease to the male part. Okay, so that is what. Now, see, color blindness is, now, there are 2 sets of genes we talked about, 2 sets of alleles we talked about. Even when you study monohybritosis, mental monohybritosis, you have come across 2 type of alleles. A gene will have 2 sets of alleles. Even in multiple allelism, we talked about there are 2 pairs of alleles will be there. That is called, one set is called dominant alleles. Dominant. For example, if you talk about capital C, it is a dominant alleles. One set we will have called recessive alleles. Very beautiful. Recessive alleles. So for example, small c, small c. So these 2 are the recessive alleles. Okay. Now, the color blindness is certain people, those who are affected with this disease, cannot visualize, cannot see the colors, especially red, blue and green. These are the primary colors. Your brain can perceive only these 3 colors. The other colors are just an imagination, that's all. What we see, only red, blue and green, your brain reads. Other colors are just seeing what our eyes is. That's all. What our eye perceives, that's all. Now, the person who is affected with this color blindness cannot differentiate or cannot see these three primary colors, red, blue and green color. They cannot see them. So, the color is always formed from a color sensitive cell called cone cells in the eyes. Eyes will have two sensitive cells. One is called rods, another one is called cones. So, rods vision in the dim light. Dark light vision is given by rods. Cones are responsible for daylight, color, brightness. So color pigment or color is perceived by the cones which is presented in the retina of the human eye. Now here, the dominant alleles, capital C, are responsible for bringing out this color pigment. Okay. Certain people, if this dominant allele is not located in a particular gene, suppose any one chromosome, and in this chromosome, this particular gene should be capital C to bring out these cones. Okay. So now what is happening? In this individual, instead of capital C, small c is there, two small c is there in recessive condition, this will not bring out these cones. So that means the person will have color blindness. So color blindness is caused by recessive gene alleles, recessive form of alleles. This much you are clear? Recessive form of allele causes color blindness. You know what is color blindness is? Color blindness is genetic disorder where a person cannot differentiate or see the three primary colors. Why? Because the cone cells are not generated. Why? Because it is having two recessive genes. Okay? And who is having this recessive gene? Female is having this recessive gene. Okay. So now where the recessive gene is located? Inside the chromosome called X chromosomes. So that means naturally female will have this kind of a disease. Okay. This much clear? Right. So now what we are doing is we are going to concentrate on two situations regarding this color blindness. We will talk two situations now. Situation number one. So now, before that, see, if you talk about XX, this is for female. Okay. So if it is having XC, C on the top, C is for color blindness. Okay. So this is called a female having disease. That means female is affected. Okay, this condition is called homozygous condition. Homozygous means same. Both are having same. This is one situation. So how you will find out female is getting disease? In her chromosome, we will mark C. C means it is disease. What is C? Color blindness. Second situation, second thing, if it is X, C and just one X is there, it is normal. Female is normal. Okay, but she is a carrier of the disease. If any male marries this girl, definitely her young ones will have the disease. Okay, so she is called carrier of the disease. She is diseased girl, female. This is carrier. In the sense for them, no disease. They are normal one. But they have the gene for transmitting the disease to another generation. Okay, that is one. Now, male like this is a normal male. Male is normal. Okay, if the male is like this, it is called, male is like this, it is called affected or carrier? Yeah, it is disease or affected. Why? Because male has only one X chromosome. He cannot be a carrier. He just can be the diseased one. Okay, so before we could study the situation, you have to understand these things. XC is for female. It is disease. Then XC and X for normal female but called as carrier. X and Y male normal. XC Y male, it is having disease or affected. Okay, right. So now we will take two situations here. We are talking about we'll talk two situations, that is the normal female. Okay, normal female getting married with a color blind man. So this is the situation. Okay, normal female getting married with a color blind man. Okay, so normal female is X and X. This is a normal female. Color blind X, C and Y. Color blind. Okay. So now F1 generation, they are getting married. So this is the gene that is located here. Okay, like this. Now what happens here? This X and this joins together. So becomes female, normal only, but she is a carrier. Okay, even a normal woman marries a colorblind man, the young ones will be like this. The young ones will be one female will be a carrier. Then join this and this. You will have normal. Male means it will be normal. If that family after getting married, if the family if she is having a male, no problem. Why? No disease. But in that family, if they are having female, normal only, but still she is a carrier. Why? If this particular girl marries any normal man, what happens? Normal. So I take this one. This is an example. This female marries any normal. See, this and this joins together gives you female, but still it is normal. Now this is male, but it is normal. Okay, normal. Now this and this joins. So gives you female but normal, but still she is called carrier. But when this and this joins together gives you male, but it is diseased. Okay, so now you become here male becomes diseased. Okay, that is the reason if the carrier female marries a normal man, it becomes a problem. In that family, if those who both got married, if male is there, nothing is going to happen. If female is there, definitely she becomes a carrier and she is going to spread the disease. So in this, how many normals are there? 3 normals. So it is ratio 3:1. Percentage wise, disease 25%. Normal 78%. Only ratios given in the book. Even they will ask you percentage wise. So 25% is affected. 75% is normal. Okay? This is clear? Right. So now, this is a parent and this is a progeny. This is also progeny. Now, talk this in different angle. Now, here, a character or color blindness is passing from last year's public compulsory 3-mark question from grandparent, GF, you remember, okay? So a character or color blindness is passed from grandparent to his grandson. Draw the flow chart for this. That is what the question. Last year's public compulsory 3-mark question. Grandfather is having a disease. Grandfather is sending the disease to the grandson. Draw the flow chart for it. So now see, this is a grandparent having diseased, color blindness. Now, remember always, very important, listen, whenever grandfather is having a disease, the grandfather will send the disease to his daughter. So through the daughter only, it will send to the grandson. See, grandfather had it. Now, who got it? Not disease, but still through daughter. And who is getting affected? Male gets affected. Okay? So from grandfather, that is the reason, if male has the disease, female is not affected. If male has the disease, male is getting affected. Okay? See, grandfather had a disease, it is through daughter. Why? Because the gene has passed to the daughter and comes here. So now see, it goes like this and comes like this. So this is called criss-cross inheritance. So color blindness is called criss-cross inheritance. In a sense, grandfather is not sending to the son directly. Okay, so this father is not sending the disease directly to the son, but sending to the grandson through daughter. So it is called crisscross inheritance. Okay, this is one situation. One situation is grandfather to grandson. Now we talk the same situation, just ulta. That is, so far we talked about the normal female with colorblind male. Okay, right. So now the same situation, just ulta, that is, we will talk about the same scenario, same scene. Now what we'll do is, here the normal male versus colorblind female. Okay, so I'll just write this colorblind female. So normal male, X and X. Sorry, X and Y. Now the female is colorblind. So both are getting affected. XC and XC. Same situation. X, H joins. So that is what it is. It is female only, but normal but still carrier. Then this is male affected or diseased. A for affected. Okay, right. So now again, this and this joins together gives you female normal but carrier. This and this male normal. Okay. Now this and this female very good affected, and this male affected. Okay, male affected. See, when female add the disease is sent through, it is who is getting affected? Son is getting affected. Female added, but who got affected? Son got affected. Then later on, daughter and son both got affected. When male add this, only another male got affected. But when female added, in first generation itself, male got affected. Second generation, male, female, both are getting affected. Here, first generation is normal. Understand? Right. So now here, see, this is the grandmother sending to both grandson and granddaughter through both. Through, this is because this is carrier and this is, okay, so it is affected. So it is again mother sends to grandson and the granddaughter. Okay, right. So this is how, if you have this one, you can clearly understand. So a normal female should not get married with a colorblind man. At the same time, a normal man should not get married with a colorblind. Okay, so this will be the problem. Okay, so this, how it's all spread? It is all spread by the X chromosome. So the disease belongs to the X chromosome, female, and female spread the disease to the male. Okay, so that is what color blindness are. Okay, so in this sex-linked inheritance, we talk what is sex-linked inheritance, we know what is sex-linked gene, then we talk what is X-linked and what is all-linked gene. Then we also discuss the color blindness in two situations. Okay, it is a very important 5 markers. Right. Question number 1. Please. Where bodies, question number 1, where bodies were first observed in dash cells of the female cat? Where bodies were first observed in the dash cells of the female cat? Where bodies were first observed in the dash cell of the female cat? Number 2. Number 2. XO female will have dash bare bodies. XO female. XO female will have dash bare bodies. How many bare bodies? XO. XO female will have dash bare bodies. Question number 3. Queen bee produces male or drones. Why the process called dash? Queen bee produces male or drones by the process called dash. By the process called dash. The process called dash. Fourth question. Fourth question. Fourth question. Who first observed the bare bodies in the female cat cell? Who first observed? Who first observed? The last question. Last question. How many number of chromosomes are present in the honeybee's female family? How many number of chromosomes are present in the honeybee's female or female honeybee? Check the answer. Question number 1. Nerve cells. Question number 2. No bare bodies. Question number 3. Parthenogenesis. Question number 4. It is parthenogenesis only correct. No unfertilized dog. I talked about the process. The fourth one, it is not Mary Lyons. 1949, Mars and Batman. They only have first observed in the. See what is the difference between Mary Lyons and Mars Batman? Because Mary Lyons has given inactive chromosome which is present in the form of euchromatin, tightly coiled. That is what discovered by Mary Lyons. Okay, but Bear and Batman, what they have told, what they have found out is they found out this bare body in the female nerve cell cat. Okay, there is a difference, right? The last one, how many set of chromosomes are located in the queen, or female honeybee is 36 number of chromosomes. That's it. So next class we will be discussing the other part of it.